So we already know that if we start off with a glucose molecule, which is a 6-carbon molecule. 6-carbon molecule, that this essentially gets split in half by glycolysis, and we end up with 2 pyruvic acids, or 2 pyruvate molecules. So glycolysis literally splits this in half. It lyses the glucose, and we end up with 2 pyruvates, or pyruvic acids. Pyruvate. And these are 3-carbon molecules. 3-carbon molecules. There's obviously a lot of other stuff going on in the carbons. You saw it in the past, and you could look up their chemical structures on the internet or Wikipedia and see them in detail. But this is kind of the important thing, is that it was lysed, it was cut in half, and this is what happened in glycolysis. Glycolysis. And this happened in the absence of oxygen, or not necessarily. It can happen in the presence or in the absence of oxygen. It doesn't need oxygen. And we got a net payoff of 2 ATPs. 2 ATPs net. And I always say the net there because you remember, it used 2 ATPs in kind of that investment stage, and then it generated 4. So on a net basis, it generated 4, used 2, it gave us 2 ATPs, and it also produced 2 NADHs. 2 NADHs. That's what we got out of glycolysis. And just so you can visualize this a little bit better, let me draw a cell right here. Maybe I'll draw it down here. Let's say I have a cell. That's its outer membrane. Maybe its nucleus. We're dealing with a eukaryotic cell. Doesn't have to be the case. It has this DNA in its chromatin form all spread around like that. And then you have some mitochondria, and there's a reason why people call it the power centers of the cell. We'll look at that in a second. So there's a mitochondria, has an outer membrane and an inner membrane, just like that. I'll do more detail on the structure of a mitochondria maybe later in this video, or maybe I'll do a whole video on them. That's another mitochondria right there. And then all of this fluidic space out here that's between the organelles. Where in the organelles, you can view them as parts of the cell that are, they do specific things. They do specific things, kind of like organs do specific things within our own bodies. So between all of the organelles, you have just this fluidic space. This is just fluid of the cell, and that's called the cytoplasm. Cytoplasm. And that's where glycolysis occurs. So glycolysis occurs in the cytoplasm. Glycolysis. Now, we all know, in the overview video, we know what the next step is. It's the Kreb cycle, or the citric acid cycle. And that actually takes place in the inner membrane, or the inner space of these mitochondria. Let me draw it a little bit bigger. Let me draw mitochondria here. So if this is a mitochondria, it has an outer membrane. It has an inner membrane. If I have just one inner membrane, we call it a crista. If we have many, we call them cristae. These little convoluted inner membrane. Let me give it a label. I'll say they're cristae, the plural. And then it has 2 compartments, right? It's because it's divided by these 2 membranes. This compartment right here is called the outer compartment. This whole thing right there, that's the outer compartment. And then this inner compartment in here, is called the matrix. The inner compartment right there is called the matrix. Now, you have these pyruvates. They're not quite just ready for the Kreb cycle, but I guess, well, it's a good intro into how do you make them ready for the Kreb cycle? They actually get oxidized, and I'll just focus on one of these pyruvates. And we just have to remember that the pyruvate, that this happens twice for every molecule of glucose. So we have this kind of preparation step for the Kreb cycle. We call that pyruvate oxidation. Pyruvate oxidation. And essentially what it does is it cleaves one of these carbons off of the pyruvate. And so you end up with a 2-carbon compound. You don't have just 2 carbons, but its backbone of carbons is just 2 carbons, called acetyl-CoA. Acetyl-CoA. And if these names are confusing, because what's acetyl coenzyme A, these are very bizarre. You could do a web search on it, but I'm just going to use the words right now because it'll keep things simple and we'll get the big picture. So it generates acetyl-CoA, which is this 2-carbon compound, and it also reduces some NAD+ to NAD And this process right here is often given credit, or the Kreb cycle or the citric acid cycle gets credit for this step, but it's really a preparation step for the Kreb cycle. Now, once you have this two carbon chain acetyl-CoA right here, you are ready to jump into the Kreb cycle, this long talked about Kreb cycle, and you'll see in a second why it's called a cycle. The acetyl-CoA, and all of this is catalyzed by enzymes. And enzymes are just proteins that bring together the constituent things that need to react in the right way so that they do react. So catalyzed by enzymes, this acetyl-CoA merges with some oxaloacetic acid, very fancy word, but this is a 4-carbon molecule. This is a 4-carbon molecule right here. These two guys are kind of reacted together or merged together, depending on how you want to view it. Draw it like that. It's all catalyzed by enzymes. It's important, some tests will say, is this an enzyme catalyzed reaction? Yes, everything in the Kreb cycle is an enzyme catalyzed reaction, and they form citrate or citric acid. Citric acid, which is the same stuff in your lemonade or your orange juice. And this is a 6-carbon molecule, which makes sense. You have a 2-carbon and a 4-carbon. You get a 6-carbon molecule. And then the citric acid is then oxidized over a bunch of steps. And this is a huge simplification here, but it's just oxidized over a bunch of steps. Again, the carbons are cleaved off. Both 2 carbons are cleaved off of it to get back to oxaloacetic acid. And you might be saying, hey, when these carbons are cleaved off, like, when, let's say, this carbon is cleaved off, what happens to it? It becomes CO2. It gets put onto some oxygen and leaves the system. So this is where the oxygen or the carbons or the carbon dioxide actually gets formed. And similarly, when these carbons get cleaved off, it forms CO2. And actually, for every molecule of glucose, you have 6 carbons. When you do this whole process once, you're generating 3 molecules of carbon dioxide, but you're going to do it twice. You're going to have 6 carbon dioxides produced, which accounts for all of the carbons, right? You get rid of 3 carbons for every turn of this. Well, 2 for every turn, but really for the steps after glycolysis, you get rid of 3 carbons, but you're going to do it for each of the pyruvates. So you're going to get rid of all 6 carbons, which will have to exhale eventually. But this cycle, it doesn't just generate carbons. The whole idea is to generate NADHs and FADH2s and ATPs. So, right here, and this is a huge simplification. I'll show you the detailed picture in a second. We'll reduce some NAD into NADH, NAD plus into NADH. We'll do it again. And of course, these are in separate steps. There's intermediate compounds. I'll show you those in a second. NAD plus, another NAD plus molecule will be reduced to NADH. Maybe we'll produce some ATP will turn to, or some ADP will turn to ATP. ADP turning into ATP. Maybe we have some, and not maybe, this is what happens. Some FAD gets, let me write it this way. Some FAD gets oxidized into FADH2. And the whole reason why we even pay attention to these, you might think, hey, cellular respiration is all about ATP. Why do we even pay attention to these NADHs and these FADH2s that get produced as part of the process? The reason why we care is that these are the inputs into the electron transport chain. These get oxidized, or they lose their hydrogens in the electron transport chain, and that's where the bulk of the ATP is actually produced. And then maybe we'll have another NAD get reduced or gain in hydrogen, right? Reduction is gaining an electron or gaining a hydrogen whose electron you can hog. NADH. And then we end up back at oxaloacetic acid, and we can perform the whole citric acid cycle over again. So now that we've written it all out, let's account for what we have. So depending on, let me draw some dividing lines so we know what's what. So this right here, everything to the left of that line right there is glycolysis. Glycolysis, we learned that already. And then most, especially introductory textbooks, will give the Kreb cycle credit for this pyruvate oxidation, but that's really a preparatory stage. The Kreb cycle is really formally this part where you start with acetyl-CoA, you merge it with oxaloacetic acid, and then you go and you form citric acid, and which essentially gets oxidized and produces all of these things that will need to either directly produce ATP or you do it indirectly in the electron transport chain. But let's account for everything that we have. Let's account for everything that we have so far. We already accounted for the glycolysis right there. 2 net ATPs, 2 NADHs. Now, in the citric acid cycle, or in the Kreb cycle, well, first we have our pyruvate oxidation that produced 1 NADH. But remember, if we want to say what are we producing for every glucose, this is what we produced for each of the pyruvates, right? This was from 1 NADH from just this pyruvate. But glycolysis produced 2 pyruvates. So everything after this we're going to multiply by 2 for every molecule of glucose. So I'll say so the pyruvate oxidation times 2 means that we got 2 NADHs, 2 NADHs right there. And then when we look at this side, when the formal Kreb cycle, what do we get? We have how many NADHs? 1, 2, 3 NADHs. So 3 NADHs times 2, because we're going to perform this cycle for each of the pyruvates produced from glycolysis. So that gives us 6 NADHs. We have 1 ATP per turn of the cycle. This is going to happen twice, once for each pyruvic acid. So we get 2 ATPs. And then we have 1 FADH2. But we're going to do this cycle twice. This is per cycle, so times 2. We have 2 FADHs. Now, sometimes in a lot of books, these 2 NADHs, or per turn of the Kreb cycle, or per pyruvate, this 1 NADH, they'll give credit to the Kreb cycle for that. So sometimes instead of having this intermediate step, they'll just write 4 NADHs right here, and you're going to do it twice, once for each pyruvate. So they'll say 8 NADHs get produced from the Kreb cycle. But the reality is 6 from the Kreb cycle, 2 from the preparatory stage. Now, the interesting thing is we can now count whether we get to the 38 ATPs promised by cellular respiration. We've directly already produced for every molecule of glucose 2 ATPs and then 2 more ATPs. So we have 4 ATPs. 4 ATPs. How many NADHs do we have? 2, 4, and then 4 plus 6 is 10. We have 10 NADHs. And then we have 2 FADHs. 2 FADH2s. I think in the first video on cellular respiration I said FADH. It should be FADH2 just to be particular about things. And these, so you might say, hey, where's our 38 ATPs? We only have 4 ATPs right now. But these are actually the inputs in the electron transport chain. These molecules right here get oxidized in the electron transport chain. Every NADH in the electron transport chain produces 3 ATPs. So these 10 NADHs are going to produce 30 ATPs in the electron transport chain. And each FADH2, when it gets oxidized and gets turned back into FAD in the electron transport chain, will produce 2 ATPs. So 2 of them are going to produce 4 ATPs in the electron transport chain. So we now see we get 4 from just what we've done so far, glycolysis, the preparatory stage, and the Krebs or citric acid cycle. And then eventually these outputs from glycolysis and the citric acid cycle, when they get into the electron transport chain, are going to produce another 34. So 34 plus 4, it does get us to the promised 38 ATP that you would expect in a super efficient cell. This is kind of the theoretical maximum. In most cells, they really don't get quite there. But this is the good number to know if you're going to take the AP Bio test or in most introductory biology courses. There's one other point I want to make here. Everything we've talked about so far, this is carbohydrate metabolism, or sugar catabolism, we could call it. We're breaking down sugars to produce ATP. Glucose was our starting point. But animals, including us, we can catabolize other things. We can catabolize proteins. We can catabolize fats. I mean, if you have any fat on your body, you have energy. You're in theory, your body should be able to take that fat and you should be able to do things with that. You should be able to generate ATP. And the interesting thing, the reason why I bring it out up here is obviously glycolysis is of no use to these things, although fats can be turned into glucose in the liver. But what the interesting thing is that the Kreb cycle is the entry point for these other catabolic mechanisms. Proteins can be broken down into amino acids, which can be broken down into acetyl-CoA. Fats can be turned into glucose, which actually could then go the whole cellular respiration. But the big picture here is acetyl-CoA is the general catabolic kind of intermediary that can then enter the Kreb cycle and generate ATP regardless of whether our fuel is carbohydrates, sugars, proteins, or fats. Now, we have a good sense of how everything works out right now, I think. Now I'm going to show you kind of a diagram that you might see in your biology textbook, or I'll actually show you the actual diagram from Wikipedia. And I just want to show you this looks very daunting and very confusing, and I think that's why many of us have trouble with cellular respiration initially, because there's just so much information. It's hard to process what's important. But I want to just highlight the important steps here, just so you see it's the same thing that we talked about. From glycolysis, you produce 2 pyruvates. That's the pyruvate right there. They actually showed some molecular structure. This is the pyruvate oxidation step that I talked about, the preparatory step. And you see we produce a carbon dioxide, and we reduce an NAD plus into NADH. Then we're ready to enter the Krebs cycle. The acetyl-CoA and the oxaloacetate, or oxaloacetic acid, they are reacted together to create citric acid. They've actually drawn the molecule there. And then the citric acid is oxidized through the Krebs cycle right there. All of these steps, each of these steps are facilitated by enzymes, and it gets oxidized. But I want to highlight the interesting parts. Here we have an NAD get reduced to NADH. We have another NAD get reduced to NADH. And then over here, another NAD get reduced to NADH. So far, if you include the preparatory step, we've have 4 NADHs formed, 3 directly from the Krebs cycle. That's just what I told you. Now we have, in this diagram, they say GDP or GTP gets formed from GDP. The GTP is just guanosine triphosphate. It's another purine that can be a source of energy. But then that later can be used to form an ATP. So this is just the way they happen to draw it, but this is the actual ATP that I drew in the diagram on the top. And then they have this Q group, and I won't go into it, and then it gets reduced over here. It gets those 2 hydrogens, but that essentially ends up reducing the FADH2s. So this is where the FADH2 gets produced. So as promised, we produced for each pyruvate that inputted, remember, so we're going to do it twice. For each pyruvate, we produced 1, 2, 3, 4 NADHs. We produced 1 ATP and 1 FADH2. And that's exactly what we saw up here. I'll see you in the next video.
Thank you for watching this video. Don't forget to like the video.